Hello and welcome to Nikolay's genetics lessons, and today's problem is which mutation should least require realignment of the homologous regions of the gene that is common to several related species. And here's a variants to choose from. And this codon table is going to help me to explain how we are going to solve this problem. Imagine that this is double-stranded DNA, but instead of two strands of the DNA, I'm going to use only one strand of the DNA, which is template strand of the DNA. Now imagine that we have a following sequence here. For example, A, T, C, and G. A, T, C, and G, and A, T, C, and G. Because code of life is read in trinucleotide sequence, so we can say that first trinucleotides code for certain amino acid, and this is going to be another amino acid, and yet another amino acid. And now let's assume that we have three base deletions. So what's going to happen? Imagine we have a deletion here. Usually the length of the polypeptide chain or protein is about 1000 of amino acids. So we just deleted one codon. We specify one amino acid out of 1000. Is it bad or not? Sometimes if this amino acid in the active site of the enzyme, for example, it's going to be very deleterious mutation, but in many cases just deletion 1 out of 1000 amino acids is not going to change how this protein behaves. So most likely this is not as bad mutation as, for example, one base deletion mutation. So what's going to happen if only one base would be deleted? For example, this base here. So what's going to happen? Take a look. Our codon sequence now is going to be as follows. So this is going to be our first codon. This is going to be our second codon, our third codon. So as you see, the sequence now is completely different. If the first time it was A, T, C for the first codon, now it is T, C, G. So completely different sequence for all following trinucleotides which specify amino acids. So after such a mutation, we are not just going to get completely different polypeptide chain, but also take a look at this codon table. And as you see, 1, 2, 3 codons here specify stop. And that means if we expect, for example, in this reading frame not to see stop codons for 1000 consecutive nucleotides, if we disrupt this sequence, chances that 3 out of 64 or 1 out of 20 codons would specify stop codon just by random chance only. So we are not only going to get completely different sequence of amino acids, but we also going to get a short version of the protein. And if, for example, such mutation happened somewhere in the middle, and the middle would be 500 amino acids, we expect that within 20 more amino acids we are going to see a stop codon, and this is going to be just very short version of the protein. Or if it's going to happen at the beginning of the protein, then we are going to get a version that is going to be non-functional, completely non-functional. So this type of mutation is very severe. So one base deletion or, for example, 4 base insertion would cause the same deleterious consequences. But now you know that 3 base deletion is not this bad. And also 3 base insertion would be the same type of mutation. Instead of 1 deletion of amino acid out of 1000, for example, we would have 1 extra amino acid in a polypeptide chain with 1000 amino acids. So probably not that bad mutation. But out of all one base substitution would be the least deleterious mutation, and take a look why. For example, compare these three nucleotides that all code for serine, and the only difference would be in the third position of the codon, and it can be any in the messenger RNA, it can be uracil, cytosine, adenine, and guanine, and still this all sequences specify serine. So if mutation would happen in the third position, it's not going to change amino acid, and we call such mutation silent mutation because it doesn't change amino acids, it's only change nucleic acid sequence. So if mutation would happen only in the last position, usually it doesn't change amino acid, so there's not going to be any change in protein, and that means in its function. In other words, we can say that 33.3% of this type of mutations is not going to change a protein sequence at all, and only in 66.6% this would lead to different amino acid. For example, instead of the cytosine here, we would have guanine. So this is definitely would change amino acid, but this is going to be a point mutation. So again, only one amino acid would be changed out of 1000, but the reading frame is going to be the same, and we do not expect early stop codons to happen in this sequence, and we do not expect completely different sequence of the amino acids, or sequence of the three nucleotides which specify amino acids. And if substitution going to happen in the third position, this is in many cases is not going to lead to any change in polypeptide sequence at all. So our choice, as you see, would be variant B, because this is going to be the least deleterious mutation for trinucleotide sequence, and thus for amino acid or polypeptide sequence. And this is all for today. Subscribe and see you in the next video. Goodbye.